These are your video notes for bacteria, and we'll start with the Archaebacteria kingdom. So Archaebacteria are prokaryotic bacteria that are, again, unicellular, just like all prokaryotes. And critically, let me get my cursor, their cell walls do not contain peptidoglycan. Peptidoglycan is a structural carbohydrate, just like cellulose. Cellulose is in plants. Peptidoglycan is in bacteria. So these cell walls do not have peptidoglycan, which is actually present in the other kingdom of bacteria, which we'll see in a second. So do not have it. Again, as prokaryotes, they have cell membranes, they have genetic material in the cytoplasm in what's called a nucleoid. Remember that there is no membrane-bound nucleus, but there is a region of the cytoplasm that has the DNA. That's called the nucleoid. And they do have ribosomes. Remember, ribosomes are not membrane-bound organelles, and they do exist in prokaryotes because you have proteins that get synthesized and translated in prokaryotes. They are motile, which means they can move. Some of them can move, and some of them are non-motile. Some of them are stuck in place. Another word for that, while we're introducing you to vocabulary, is sessile. Sessile just means that you're stuck in place, you can't move. So for instance, plants, like the trees outside, are sessile. They do not move. So some can move and some can't. Some make their own food. They are autotrophic. Auto means self and troph means feeding, so self-feeding. They can do photosynthesis because some have chlorophyll in their cytoplasm. And others are heterotrophic. They have to consume the remains of other individuals or organisms to get their food. These are extremophiles. They live in extreme conditions. So they can either live in very, very salty places, very, very hot places, very acidic places, very, very low oxygen places. But they're everywhere. They're like on you, they're in the classroom, they're in your house, but they are also, especially in these very, very extreme places. They make methane, so they're called methanogens, and we believe they are our first organisms on Earth because, well, first of all, they are simple prokaryotic bacteria, but also they can survive in early Earth conditions with low oxygen or no oxygen, either very, very hot, high in sulfur, high in salt, etc. So here's some examples. The salty pools of the Great Lakes and the Dead Sea contain Archaebacteria. The hot springs and geysers of parks, remember the DNA polymerase that we use for polymerase chain reaction actually came from bacteria from Yellowstone's hot springs. Guess what? Those bacteria were Archaebacteria. Black organic mud at the bottom of bogs or swamps with no oxygen or very, very little actually can and do have Archaebacteria, as do things like the stomachs of cows and sheep and deer and other ruminants that are a major source of methane. So these are all locations where Archaebacteria can live. Extreme environments. Here's another example here. Deep ocean vents is the leading candidate right now for the origin of life on this planet. There's basically what we think is happening here is I'm not going to talk about it. Chemosynthesis. Organisms still have energy at the bottom of the ocean because they get energy from the chemical bonds of chemicals. There's a lot of sulfur in here, which is a major component of amino acids. So you guys can Google chemosynthesis and deep sea ocean vents and origin of life. There was a video on it last unit too. That was a supplemental extra video. Go ahead, check it out. But this is one of our leading contenders for origin of life on this planet. Kingdom Eubacteria is also a bacterial kingdom, but this is the bacteria that you usually think of when you think of bacteria. They are also unicellular. Their cell walls do contain peptidoglycan, so this was a major difference. We didn't realize there were two bacterial kingdoms until we discovered this peptidoglycan difference, and actually they used to be called the monorens before this was discovered. Monorens, or the monarens. There was a kingdom called the Monera Kingdom or the Monera Kingdom that we no longer use because we discovered this difference and other genetic differences between these two. So this is a second bacterial kingdom. It's therefore also prokaryotic. Some are autotrophic, like blue-green algae, and others are heterotrophic. It just depends on your species. Again, some are motile and some are not. Some have flagella, some have little cilia-looking things where they kind of spin around, and others are stuck in place. I mean, strep, if you've ever had strep throat, E. coli, salmonella, lactobacillus is a bacteria in yogurt. These are all eubacteria. Here's the general shape of bacteria, both archaea and eubacteria. Again, the only difference is whether or not they have this peptidoglycan, but the overall structure is going to look very similar. You have your cell wall, including a cell membrane. You've got ribosomes, you have the DNA in a region called the nucleoid, even though there's no membrane-bound organelle around it. Maybe they have flagella or multiple flagella for movement, and pili actually for reproduction, which we'll talk about in just a second. Shapes of bacteria include round, rod, and spiral. So coccus for round, so streptococcus, strep throat, is a circular round bacteria. Lactobacillus, for instance, is a rod-shaped bacteria in your yogurt. And spirillum are spiral-shaped bacteria. These are usually not in foods that you eat. But those are your three shapes: coccus for round, bacillus for rod, and spirillum for spiral. For reproduction, bacteria reproduce very quickly. They have rapid generation times of as quickly as 20 minutes in ideal conditions. And they reproduce one of two ways for the most part. They can reproduce by binary fission, which is basically a form of mitosis. I'll say form of mitosis. It's not exactly mitosis, but it's incredibly similar. And it's basically you're making a clone of yourself, and for all intents and purposes, that's really what it is. It's mitosis. And conjugation is a form of sexual reproduction. So again, binary fission here is asexual. The cell is going to divide in half, producing two identical daughter cells that are clones of each other. I mean, it is mitosis. It's just called binary fission because there are some slight differences. You don't have to know the differences, but do know that it's called binary fission. Just, I guess, while we're at it, bi means two, and fission means to split. So you're splitting in half. You're splitting in two. Conjugation is bacteria sex. It is sexual reproduction. So this is the pili that we were just talking about. Bacteria can construct these bridges, these pili, between each other, and it's not wide enough for bidirectional flow. So there's one giving and there's one receiving bacteria. And usually what they send to each other are the circular plasmids. This is what usually happens. That plasmid is copied, and the copy is sent to the receiving bacteria. So it's a way to increase genetic diversity among bacteria. And again, that bridge, that link between the two is called a pili. And you can do this with multiple bacteria. There's 3 bacteria having this done here. So it's not just between 2. You can have whole groupings of them. All right, so why do we care about bacteria? Well, the first is their role in the ecosystem. They are decomposers. They break down organic compounds into simpler, smaller molecules. If you ever take AP Environmental Science or an environmental science course in the future and you ever visit a water treatment plant, they are used very widespread in water treatment plants and are actually a necessity in cleaning water and removing different compounds and atoms and nitrogens and stuff. And there are these massive settling tanks, and they're full of bacteria and stuff. So they're incredibly useful, and we use them every single day. Nitrogen fixation is a process where gaseous nitrogen, the gas that you breathe, the gas that makes up most of our atmosphere, gets converted into a usable form called ammonia and ammonium and its derivatives. Because nitrogen gas is inert, it's not very reactive, and it doesn't It's not usable by a lot of plants. So bacteria are actually one of the organisms that can quote unquote fix it. This is called nitrogen fixing. And I'll show you a picture in a second. We use bacteria in food. If you ever have fermentation or if you're making yogurt, for example, you can use the fact that they produce lactic acid to help you make food. And yogurt, pickles, sour cream, and kimchi are all produced because bacteria do fermentation, and this lactic acid is critical in making these types of foods. All right, so here's the nitrogen fixation example. If you look at the roots of legumes, legumes are bean plants, and you'll see little nodules that are colonies of bacteria, right? So these bacterial colonies, if you zoom in, this is what you're looking at over here on the right, you can see all the individual bacteria, but they are fixing nitrogen. They're taking gaseous N2 and they are fixing it or converting it into a usable form, NH4, NH3, nitrates and nitrites, right? So bacteria do this. It's a symbiotic mutualism, which we'll talk about next unit when we do ecology. Both the plant and the bacteria benefit. The plant gets usable nitrogen and the bacteria gets a home. All right, here's a picture of lactobacillus. You can look on the back of any yogurt container and there should be like a list of bacteria that's in there. What shape is this? Well, it's in the name. It's a rod. Right, so what you'll see is that in the genus of a lot of these bacterial species, the shape of the bacteria is part of the genus name, right? And then lacto is from lactose. It's the sugar that is broken down by these bacteria. So these are rod-shaped bacteria that break down lactose, lactobacillus. And they produce lactic acid. And that's part of the species name. All right. Remember that, again, some of these are autotrophs, which means they can do photosynthesis, but remember that they are prokaryotic and do not have chloroplasts. So where is the chlorophyll? Well, the chlorophyll floats around in the cytoplasm, right? But they do photosynthesis just like some plants can. So these photosynthetic bacteria are your blue-green algae, also called your cyanobacteria. Right? So you do have photosynthesis evolving before plants, and that's a key feature of the evolution of life on this planet. Plants were not the first to evolve photosynthesis. That was done by the cyanobacteria. And the other thing is that the cell membrane can double as a membrane for a lot of the chemical reactions that eventually are specialized in the chloroplast. So it's a combination of the chlorophyll in the cytoplasm and the membrane, the cell membrane, as a surface for reactions to occur on. We're going to end here with some bacterial diseases. Bacteria cause diseases in one of two main ways. They literally damage the cells themselves. They consume the cells for food. Tuberculosis is an example of this one. Other bacteria will release toxins that irritate tissue and skin and surfaces like strep throat. So one of those two things is probably going to cause you to feel ill or develop a fever. That's your body reacting to the bacteria and trying to make it uncomfortable to live there. There you go. So those two. Here are some others. These are, it's not an exhaustive list, but these are some of the ones you're probably more familiar with, right? Tooth decay, Lyme disease. Yes, the ticks carry it, but it's caused by the bacteria. Tetanus, there's the tetanus shot for the rusty metals, right? If you ever have kind of cut yourself on like a rusty can or something, you have to get a tetanus shot. It's to prevent the Clostridium bacteria. Tuberculosis, salmonella in a lot of your meats, pneumonia, cholera, and water supplies of like the 1800s and stuff, right? So these are all bacteria and diseases that they cause and how to treat them. So to prevent bacteria or bacterial diseases, you can have a vaccine, right? here, I mean, there are vaccinations for tuberculosis, and there's a tetanus vaccine, right? Before you get infected, you expose the body to a damaged or killed version of the bacteria, a weakened version, and your body learns to defend against it. Just like a viral vaccine, it can make antibodies against bacteria. So vaccines are preventative. Once you have a bacterial infection, you can use antibiotics. It literally means against life, antibio. So these are some kind of chemicals that kill the bacteria, prevent it from reproducing. They don't work on viruses. They only work on biotic things, living things, so only bacteria. They can disrupt the cell wall. They can disrupt the bacterial ribosomes, but they prevent reproduction and they kill the bacteria. Penicillin, ampicillin, amoxicillins, there's actually very common allergy to these groups of medicines that are all kind of derived from each other. You might have taken a Z-Pak or things like that. Those are all antibiotics. Important to take the whole course of antibiotics, because if you don't, you run the risk of selecting for resistant bacteria, and then you can get things like MRSA or gonorrhea that are very resistant to our current antibiotics. So please take the whole pack as instructed, because you don't want to be artificially selecting for bacterial resistance in common bacteria. It's evolution in real time. All right, disinfectants, the last slide here. Disinfectants obviously are things, Lysol soap. You guys know this. Again, disrupting the cell wall, usually lysing and breaking the cell open so that it's basically dead. Pasteurization, Louis Pasteur is the scientist that developed this. That's why it's named after him. When you heat, usually liquids, or you can actually pasteurize eggs and things like that too. But you initially heat them to kill bacteria, and then you cool them back down for consumption, right? So usually you'll see things like orange juice or milk or yogurt is pasteurized. It's been preheated to kill bacteria, right? Not enough to cook it, but enough to kill bacteria. And then refrigeration. Remember, refrigeration is low temperatures. You're not going to kill bacteria from refrigerating them, but you will reduce enzyme activity, right? Remember, the enzymes have very, very narrow windows of temperature and pH that they function at. They have optimal temperatures, right? So dropping the temperature down in a refrigerator, for example, slows down the movement of enzymes. It slows down the chemical reactions going on in the bacteria and limits bacterial growth. So you can preserve food in refrigerators for this reason, and it's also why if you leave food out on the counter at room temperature, you probably don't want to eat that because it's probably going to be full of bacteria, and you risk getting sick at that point. So let me know if you have any questions. I hope this helped, and I will see you in class.